What I find interesting is the interaction between scientists working in research centers, and such as the LMB, and companies developing detectors or microscopes, for example. However, the companies and research centers might have different objectives, and the development of the cryo-EM would not have been possible without such collaborations. So can you tell us a bit about mutual benefits and interests and how these collaborations are developed, managed, and sustained? And are there any shared patents?
Yes, all of these things. So, clearly, in any field, there are new discoveries, and usually it's the people in academic or research institutions that are making the basic fundamental discoveries. And it's all done with one-off, with prototype instrumentation developed. And they make it work, and they get the results, and then they publish the papers. And the skills that you need for doing that type of work are completely different from what you need to manufacture 500. The philosophy of most university departments, research institutions like the MRC Molecular Biology Lab, is that any new development, whether it's protein sequencing, DNA sequencing, protein crystallography, electron cryomicroscopy, and so on, the idea is to develop the first one or two prototypes, and then you spin it out into a company, either a small company or it gets licensed to a big company. And that's been happening certainly in our lab, but in most academic labs for 50 years or something like that. And so Ernst Ruska, when they developed the electron microscope, it was the early German companies, Siemens and Zeiss, that developed it. There were at one stage 20 companies making electron microscopes in the world. Then obviously competition has dropped it now. There are only 3 doing the really expensive ones, but there are many others doing small ones. So usually, as soon as something has been a success and 1 or 2 papers have been published, there is now in most universities or research labs, you have a technology development or technology transfer part of the organization. And in round about 1990, for example, when we were starting to see them, the MRC wasn't so advanced in that. There were maybe 3 or 4 people that gave advice about patents. And usually you would patent something, and then the patent would be licensed, and then a company would take it up. But because of, let's say, government encouragement and the need to get more money in, these technology transfer departments have grown in size. And so now all of the MRC inventions, patents, royalties, and so on, company startups, is done with a group that used to be called MRC Technology, and it was wholly managed by the MRC, but it was a charity itself. So that has now been separated, and all of our patents and company startups and so on is done through a company called LifeArc now, which has about 150 people, so not 4 anymore, 150 people. And they have, from this lab alone, started about 20 companies. And usually the scientists that are involved in making the fundamental discoveries, they are often the founders of the company. So they would begin by spending part of their time. For example, we had one scientist, Jonathan Carn, who had an idea for a company called RiboTargets, and he spent 5 years continuing his work for the MRC, 50% of his time, and the other 50% he went to a company on one of the science parks near Cambridge to start the company. And then that company has gone on. There's been a lot of companies like this. The most successful one has been Cambridge antibody technology that Greg Winter started. That was then taken over by AstraZeneca. So usually these small companies, they grow to a certain stage and then they get bought. They could go on. One of the biggest ones, Celltech, was one of the first companies started by the MRC. That's now part of a Belgian company called UCB Pharma. For example, with the detectors, we were involved in developing one of the detectors as an academic consortium in collaboration with a group at the Rutherford Appleton laboratory that's co-located with the diamond synchrotron. And so they spent from about 2003 to 2008 developing a prototype direct electron detector. That was then licensed to the company that's now Thermo Fisher. It's called Falcon 1, 2, 3, 4, and so on. And so I think the sensors still come from the same foundry that made them before, but it's spun out so that you have service engineers, salesmen. There's a whole infrastructure of companies. So I think there's a lot of synergy between the discovery end and the development end, and then the let's say providing a service end to it. And so now I think Thermo Fisher, they've sold about 150 of these high-end Krios microscopes, and much of the technical development will have been done inside the company, but many of the components, for example, the field emission guns, the detectors, there are face plates that give you better contrast and so on. These are all academic developments that have been folded into what is a commercial product. And then you do need some competition. Otherwise, if you only have one company, the price, as if by magic, continues to go up. And so when it gets too expensive, people say, I think we should perhaps start another company. And then so I think it's not different. Scientific research, scientific instrumentation is not different from, my view, is not really different from selling bread or toothpaste. They're products eventually, and then you have to make them work well. Like the new cars nowadays are far, far better than the cars you would buy 50 years ago. Much more reliable. They're much more efficient. They're much more pleasant to drive. So the same is true of the scientific instruments, the microscope. And you need it to be commercialized because you get the economy of scale. Whereas academics, they make one, and by the time they've made the third one, they're already bored, and then they're not doing it very well, and it's expensive too. So you need the efficiency. So I think most academic departments or research labs now have upfront, clearly stated policies. The scientists are encouraged to do their research, publish their papers, but they're also encouraged, if they have a bright idea, to spend a bit of their time developing it so that it becomes a greater benefit to a wider number of people.
Can you tell us a bit about your activities as entrepreneur?
Okay, so I wasn't specially personally involved in entrepreneurial activity, but in the 1970s, the philosophy then was the academics in the research labs, we should just do our work, publish our papers, and then someone else will read them, and then they will start a company. But in 1975, Cesar Milstein and George Kohler discovered this way of fusing antibody cells from a mouse or a human with cancer cells. And so they made fused cells, they called hybridomas, which produced forever one pure antibody. And they said in their paper, they published it in Nature, they said, they thought this might be quite useful in diagnostic or therapeutic medicine. And they did try to patent it, but they were told that there's nothing about it that should be patented, you should go ahead and patent it. But later on, it turned out to be very important. And so now it turns out that most of the highest effective and new and expensive drugs on the market, almost all are these hybridomas, but with further developments that have happened. But so when it became clear that this was going to have a major impact on industry and pharmaceutical, the MRC was criticized back in the 1980 for not having patented it. Then the culture changed from one in which everyone published it and got on with their work and someone else did the development, to encouraging people explicitly, the scientists who work here, to, alongside their normal work, to patent things, to suggest, or maybe even to start their own company. The first one was Celltech, about 1982. which commercialized diagnostic monoclonal antibodies against all the human blood group antigens. And that grew lots of people from the LMB. 2 people who were group leaders resigned their jobs and went to be chief scientific officer and business officer and so on, running the company. And then many other people went there as technicians and so on and so on. So it grew, and that still exists as UCB Pharma. That's still part of their drug. But that then brought in the idea that government-funded labs ought to take an interest in development, exploitation, company startup. So since then, there's been about 20 companies, maybe more, just from LMB itself that have been, and then probably more than half of them have developed into quite successful companies. And for example, Greg Winter's he started 3 companies. So Cambridge Antibody Technology, Demantis, and there's another new one called Bicycle Therapeutics. At one stage, another colleague, Chris Tate, and I, we had been working on bacteriodopsin, like you see. And then it turned out that there is a very big family of human proteins, about 400 of them, that look just like this. They all have 7 transmembrane helices, and they're in the human body. They are responsible for vision. So the red, green, blue color pigments, the dark vision, rhodopsin, 4 of them are in the eye. And then in your body, things like in your heart, you have the adrenergic receptor. So when you reduce adrenaline, your heart beats faster. That is a protein just like this. Adrenaline binds to it, signals to the cell. And there are another 400 that do different physiological processes. So when that became apparent, and that wasn't clear until about 1985, when the first, so beta-adrenergic, 1985, turned out to have a similar sequence to rhodopsin in the eye. That was the first 2. And then there was the human genome about 20 years ago. That revealed another 400, and then 400 all the factors. So it's a big, big family. So we decided we better work, since medical research, we better work on this. Because our methods and our technology. So we worked from 1990 on G protein coupled receptors. And I actually only did a little bit myself, but we had a group that gradually grew in size. And the group, so Gebhard Schertler, Jade Lee, they solved the structure of rhodopsin, which was the visual pigment in cows. It was not cows rather than humans, in the 2000, 2004. But it was very difficult to get the structures for these hundreds of other GPCRs that are very important in drug development. And so at one stage in 2005, we hadn't succeeded, and obviously it was of some value. So we got a grant from a pharmaceutical company called Pfizer, 50%, and another grant from MRC Technology, that's now LifeArc. And they funded 4 people to come and work on our ideas for doing G protein coupled receptors. And remarkably enough, it worked out really well. So after we'd been doing that for 2 years, that was Chris Tate and I, and we said, well, what shall we do now? Because the grant was a 3-year grant. Shall we get another grant? Shall we have a public sector consortium where everybody's allowed? Or should we start a little company? So we said, oh, well, maybe we should start a little company because we were supposed to be doing that. And so we knew another scientist who had been in big pharma and little pharma and so on, called Malcolm Weir. And just at the moment, we were thinking maybe it would be a good idea to start our own little company. Malcolm Weir, his position as the chief executive officer of a company called Informatica, he was sacked, he was terminated, and so he was unemployed. So we said, oh, well, maybe not only should we start a company, but Malcolm could be the... So MRC Technology said, oh, that's a good idea. So they hired him. They paid him for 6 months as a consultant. And his job was to go and get money to start a company. And within 2 weeks, he said, oh, I've got £1 million. So he got it from venture capital or something. So anyway, that went on, and now the company is called Heptares. It's been bought by a Japanese company. It's called Sosai Heptares, 150 employees, and is doing lots of drug development. And we've all been bought out. MRC got some money out of it. And so it has been translated into something useful.